I want to thank you very much for inviting me to speak here today. Um, like I do usually with my talk here, I'm not always sure what my audience is for for quantum biology. So just so you know that my presentation is assuming there is a mixed audience with a diverse background. So please don't feel insulted if I am explaining something that you find very basic. And if it's physics and I explain wrong, I will happily take the correction. Just know that the basicness isn't aimed at you. So the title of my talk today is Weak Magnetic Fields Manipulation of ROS Signaling During Regeneration. And my graduate advisor told me to begin my talks with the ending part, the conclusion. So basically, what I'm hoping that I will show you is that our work has demonstrated that weak magnetic fields in a field strength dependent manner can be used to modulate stem cell mediated growth in an organism by manipulating superoxide levels, at least in our organism. But to get everyone back to a place where you can sort of appreciate this statement, or at least why I am making it, we'll go back to the beginning, which for my lab is the planaria, a non-parasitic flatworm. Here they are. Freshwater, not parasitic. You find them in freshwater. You can find them basically anywhere in the silts of your favorite sort of lake bed or stream. And they're very easy to care for. As you can see here in the lab, we keep them in Tupperware. And the adults, even of the same species in the same colony, can vary in size, and we'll explain why later. But typically they range from a few millimeters to a few centimeters, depending on the species. They have several characteristics that make them awesome from my point of view for research, not the least of which is a complete central nervous system with a true brain, which you can see this sort of V-shaped structure in green. And just like humans, the brain is bilobed. Interestingly, it's thought that this looks similar to the very first central nervous system with brain that evolved. It's really good for evolutionary studies as well. Planarians also have, and this is why we're super interested in them, an exceedingly large population of pluripotent adult stem cells. So we work with adults, not embryos. And what you can see here in green is the dividing cells that we have labeled. And these aren't all the stem cells. These are just the ones that are actively dividing at the time that we took this picture. In fact, it's about 25 to 30% of all of the adult cells are stem cells in planarians, which is kind of astounding if you compare only about 1% of your cells are adult stem cells. So it makes them really good for studying stem cell biology. Now, planarians are hermaphroditic and they can reproduce sexually, but frankly, we're not really that interested in them. We're interested in them because they can also do this, which is reproduce asexually, which occurs through transverse fissioning, which is just a fancy way of saying that the worm sort of pulls itself really, really thin until it rips apart into two pieces. Whole process takes about 45 minutes, and both fragments are going to go on to regenerate into complete worms. So if you look here, the head fragment on the left is going to regenerate a new tail, which it's in the process of doing, that white part. And the tail is going to grow a brand new head with a completely brand new brain. Pretty amazing. So we basically co-opt this ability, and this ability to fission and produce two new worms from a larger one is one of the reasons why our adults are not all the same size, because you never know when they're going to fission. But we use this ability, their natural ability to regenerate, to sort of study how the regenerative process occurs in the lab. So if you decapitate a worm, which is basically what I'm going to show you throughout my talk, sort of like they have done here, instead of dying like a sensible creature, the worm actually initiates the growth of new tissue, which you can see here labeled in white. It's called the blastema. And fortunately for us, the pigmentation is the very last thing that regenerates. So it's fairly easy for us to tell what is new tissue. Now, the blastema is a special tissue. It's a collection of unpigmented, undifferentiated progeny, by which I mean they're not mature cells. They don't actually have a specific function like producing stomach acid or being a muscle, that sort of thing. And it arises, this blastema, as the result of stem cell proliferation at the wound site. So in this picture, if you can sort of see it, the dotted red line is the wound site, and the stem cells themselves are in blue, and they're dividing right near the wound site. And the progeny, the earliest progeny, the descendants of the stem cells that have been dividing, are in green. And then over time, they start to differentiate into more specialized type of progeny, but still not mature cells, which we see in red, so early and late progeny. And it's these progeny or descendants of the stem cells that create the blastema and ultimately are going to differentiate into different types of mature functional cells. So the blastema is going to go on to form the basis of the missing tissues, although some of the original tissues also get rearranged, but I don't ever really talk about that during quantum biology. Anyway, the really interesting thing is that within sort of 7 to 14 days, the worm is able to regenerate a brand new head. So what you have here on the left is a whole worm with sort of red lines where I did the cuts. And then all what you're seeing on the right is the exact same worm or piece of worm where I took that middle and we followed the middle fragment of that worm over about 17 days. And you can see that it eventually makes a brand new planaria, just a little smaller. Interestingly, the brain primordia start within 24 hours, so it's very quick. And the eyes and the brain are completely functional within 7 days, even though this piece didn't have any brain to start with. And overall body shape, which is something my lab is very interested in, is restored within 2 to 3 weeks. All right. So why is a stem cell planarian researcher interested in quantum phenomena? Well, the link comes from cell signaling downstream of radicals, which I might about to be, I don't know, what's the biology version of mansplaining? Biology explaining to a bunch of physicists. But if I remember my high school physics, which I didn't at first, electromagnetic fields are present everywhere, both as the Earth's natural geomagnetic fields, which averages 25 to 65 microtesla, depending on where you're standing, as well as around from the technology around us. Now, strong magnetic fields are well known to affect chemical reaction rates and free radical concentrations in cells. And as we all know, diagnostically, they can be very useful, as in magnetic resonance imaging or the MRI seen here. But MRIs have magnetic fields of 1.5 to 3 Tesla, which is more than, in some cases, a lot more than 30,000 times greater than the geomagnetic field. So what about weak magnetic fields, which in our case, we're calling any field that's less than 1 millitesla. So we're in the microtesla range. It's been shown that weak magnetic fields also can affect cellular processes. So for instance, in cell culture, which is what we see here with rat skeletal muscles, it's found that myotome or muscle cell formation and growth occur much faster in magnetic fields that are slightly higher than Earth normal. Here they have 160 microtesla. Similarly, if we look in the other direction, they've reported in a cancerous cell line that is exposed to magnetic fields less than 18 microtesla. So now we're less than the geomagnetic field, that the cell growth of fibrosarcoma, HT1080 cells, if you care, is decreased. So I wouldn't take these directions like bigger than Earth normal being positive for growth and lower than Earth normal being less, though sometimes it works that way, but our work has shown that it's likely to keep cycling binary. We'll talk about that in a minute. All right, so we also know that certain specialized cells have the ability to sense quantum phenomena, so such as the migration of birds in magnetoreception and photosynthesis, which is basically photon sensing. But our question was, what about other types of cells? And how would that work? So the model that we deal with is through spin dynamics and the radical pair mechanism, which if Clarice is there, she's talked about this, the spin dynamics. But in summary, just in case, radicals are basically molecules with an odd number of electrons, and they thus have an unpaired electron that can be in one of two spin states, basically up or down, which importantly, from our point of view, affects molecular binding. Whether the spin state is up or down will affect whether it can or cannot recombine with other molecules. So in this way, magnetic fields can drive spin states, either a singlet state where electrons have an opposite spin state. So up here, if one's up and one's down, molecules can bind. That promotes recombination. And this is going to reduce free radical concentrations, because a free radical is a molecule that is lonely and looking to cause problems because it just has a single electron in its valence orbit. Okay, so but different magnetic fields can also sort of produce a triplet state where electrons have symmetrical spins. So this, from our point of view, is important because it can promote diffusion. And this is where we can increase radical concentrations, prevent, even at the millisecond time span, which would be forever for a cell, and cause more radical concentrations in the cell. So you can see the theoretical model. We can either increase the amount of radicals or decrease the amount of radicals. And this is significant because there's one class of free radicals. There's definitely more than one, but the ones we're talking about today are reactive oxygen species, or ROS, and they're very important signaling molecules that participate in redox biology. In particular, we are interested in the species hydrogen peroxide and superoxide, though they're definitely not the only radicals. But these two in particular
Wendy,
are, yes.
Sorry, Wendy, your internet was a little bit unstable. Can you go back to your slide when you were talking about triplet recombination and increased levels? Yes, we lost you there.
Okay, I am sorry. Is this better now?
Yes.
Okay, good. I have no idea what happened. Okay. So all I was really saying with the triplet state, right, is that if they're in the same direction, whether they're both up or both down, it's going to prevent, from the molecular point of view, molecules from recombining, and this is going to increase the amounts of free radicals. And from the biologist standpoint, basically the take home of this model is that this is a method by which weak magnetic fields can either increase the level of the amount of free radicals in a cell or decrease it, which is very significant to us because there is a class of free radical. There are several, but the one we're going to talk about is reactive oxygen species, or ROS. These are, of course, important signaling molecules that participate in redox biology, but in particular, we are very interested in hydrogen peroxide and superoxide. There, as you can see on the left, there are several ROS species, but the ones that are best studied and known to participate in cellular signaling pathways, things that control cell behavior. So the thing to know about ROS signaling is that it works in a threshold dependent manner where either too much or too little can lead to disease or cell death. But intermediate amounts, some random in-between amount that we don't have a good handle on, is well known to regulate cell signaling. It controls a lot of stuff that goes on during development and embryogenesis. It controls things in regeneration. It can control changes in gene expression, control cell migration and cell adhesion, a lot of very important cellular processes. More importantly to my research is that levels of ROS are known to regulate stem cell activities, what a stem cell chooses to do, such as proliferation, in other words, cell division, and it can control, because it can regulate that, it can control stem cell divisions to self-renew. Is it going to divide and make more new stem cells? Is it going to divide and differentiate into mature mature cells, or is it going to do both? Or even remaining quiescent, which is a fancy word meaning to do nothing, but to maintain your stem cell population for the future. So this is a very important signaling pathway. And I'm not really going to talk about that today, but basically, we know that ROS signaling is required for regeneration in many different model species and in different types of organs. So briefly, what you have here on the left are tadpole tails, where ROS is at the bottom in white. Zebrafish fins, where you're looking at the effects of loss of ROS in the middle on the right. The middle right, anyway, and axolotl salamander tails. And all of these are showing that if you lose ROS, it leads to a lack of new regenerative growth. So you need the ROS for regeneration to occur. So what about during planarian regeneration? We went through to figure out what ROS signaling is doing in planaria, since it's our model organism. And we found that the first thing that happens is about 30 minutes to 1 hour after injury, there is a large spike of ROS accumulation at the wound site. So what you're looking here, sort of kind of can't see, is we're looking at a general ROS labeling dye, which we're showing as a heat map, where sort of this white, red, greenish indicates relatively more ROS, and the blue and black, if you can even see it, represents a lack of ROS. And we're looking at decapitated worms, and this is 15 minutes to 45 minutes after we cut the heads off, where we find that this spike of ROS occurs at the wound site. So that's very early. Now, during this same time frame, wound closure occurs to prevent tissues from leaking out. It's one of the first things that happens in most wound situations, where you need to get a very quick patch seal of cells over the wound so you don't lose all the stuff on the inside. And we know that ROS is involved in this as well, but I'm not going to talk about that today. But this sort of initial ROS spike is concurrent with actually closing the wound. All right. So what happens after that first hour? At around 4 to 6 hours after injury in planaria, there is a response of stem cell division across the entire animal, not just at the wound site, which you can see here in red on the right, where the entire stem cell population responds by increasing proliferation and migrating to the wound site. All right. This is because we've done something very severe. Now the cutoff head is to the left, and that's why we get such a big stem cell response, because we've basically cut off its brain. So, all right. So 6 hours, the end of this part, is also when wound healing phase is considered complete. And that blastema that I talked about, formation really is thought to start to occur. Blastema formation, that new tissue, the white tissue you can see here on the right, that formation sort of peaks around 72 hours, or rather day 3. Conveniently, we're going to be able to see that throughout here as the white tissue. So we've got an outlined, basically a time frame of what happens during regeneration, where the order is injury, then ROS accumulation, then stem cell proliferation, then new tissue growth. All right, so it was a lot to throw at you. What's the point? So here are the pieces, right? We've talked about how weak magnetic fields can affect spin dynamics, which are able to change free radicals, and one of those radicals is ROS. We've also talked about how ROS and signaling downstream of it, and like gene expression changes, that sort of thing, can control stem cell activities. And then we also have explained how, after injury, ROS accumulates in our model system and several others, or most others that have been studied actually, and leads to new tissue growth. So the evidence suggests if we link all of these things that have been shown in the literature together, that weak magnetic fields could control tissue growth during regeneration in the planarias. So that's where I come in. All right, so we basically asked a seemingly very simple question, which is, can weak magnetic fields be used as a tool to control stem activity during regeneration? So here you see a picture of the mu metal shield box and apparatus that we use to control magnetic field exposure in our experiments. We call this the mag shield box, and the mu metal box blocks magnetic fields. On the left is a picture of the inside of the box with the partition, and on the right are the Helmholtz coils that we place sort of in the center of each partition so that we can experimentally produce a static weak magnetic field of a given strength. There's a whole bunch of literature on frequency and different hertz, but we've not even gotten into that can of worms in our lab yet, though we're starting. We typically run an Earth normal, which for us is 45 microtesla on one side of the mu metal partition, at the same time that we run an experimental field strength, whatever that is, on the other side. All right, so what did we do? We took those trunk fragments, we can see here on the left, where we just cut off the head and tail, and wanted to see what happens when we expose them to different weak magnetic fields. And as you can guess, since I'm talking about it, we found that exposure to static weak magnetic fields is actually able to regulate regeneration in planarians. So we looked here at the top. What we're looking at is at 3 days post-injury, when that blastema size is the greatest, and we exposed them to a range of field strengths from almost 0, about 1 or microtesla or less, to 900 microtesla. And then we sort of quantified what the blastema sizes are. And as predicted by the radical pair mechanism, where the spin dynamics say that they can change in either direction, both promoting or inhibiting free radical formation, we found that different weak magnetic field strengths cause opposite effects on tissue growth. So some here in red, guess if we're red-green colorblind, this isn't going to help, sorry. The down ones, which are in red, were found to inhibit tissue growth, and we had some green ones opposite that caused growth. So for the rest of this talk, I'm basically going to show you examples of each of these strengths, which for inhibition was 200 microtesla, and which for activating is 500 microtesla. So our controls are going to be in the middle from here on out. And what you see is that if this is a normal amount of blastema size, what we get is a reduction at 200 and a significant increase in the amount of tissue, which is very interesting to us, with exposure to 500 microtesla. Now, again, we're saying that we think this is due to ROS. So if we did the same thing, expose them to the same range of field strength, 0 to 900 microtesla, but in this time looked at ROS levels much earlier, 1 hour post-injury. So this is only an hour after we've amputated them. Again, we see the same thing. We see ranges that will decrease the amount of ROS and increase the amount of ROS, with 200 almost not completely, but basically in this assay, inhibiting ROS formation at the wound site, and with 500 microtesla significantly increasing the amount of ROS. So because we're molecular biologists and we sort of want to know the mechanism, we went through to sort of see how this was working, because getting from ROS an hour after injury to a bunch of new tissue leaves a lot of stuff unsaid in the middle. Shorthand, what we see is that ROS causes a change in the amount of this gene, heat shot protein 70, after injury. So 200 inhibits its expression and 500 increases its expression. And this is important because this HSP70 is required to signal to the stem cells to divide. So if we look at stem cell division directly, not just as a measure of how much growth there is, we can see that at 200 microtesla, we get significantly less dividing cells. And at 500, we get a significant increase in the amount of dividing cells. And if we parse that even further, if we look at the actual overall number of cells, stem cells, so this in red here now is not just the dividing stem cells, which is what we saw before, but all of them, you can see that there's an overall net gain or loss depending on field strength. And if we go and look at the progeny that are in the blastema, we see the same pattern. 200 blocks the number of progeny and 500 increases the amount of progeny, which makes perfect sense if you're increasing the amount of stem cell division that's going on. So basically, if we follow the whole connection of how we get from ROS to new tissue growth, we see, here's the quantification for these last two slides, if you care. Showing that weak magnetic fields can either significantly inhibit, in this case at 200 microtesla, or significantly increase 500 microtesla in wound site ROS signaling that controls the stem cell response after injury, which is really interesting and has a lot of exciting implications. But again, developmental biologist, I basically kind of want to know what ROS signaling pathways are being affected. What exactly is weak magnetic field exposure affecting specifically? So if we look through the literature, one of the consensus pathways for ROS, if you start with molecular oxygen, you can see that superoxide here can be converted into hydrogen peroxide by the enzyme SOD. This is superoxide dismutase. But basically in a single pathway, we can get from molecular oxygen to superoxide to hydrogen peroxide. Both of these species that I said we were going to be looking at, the superoxide and the hydrogen peroxide, are known to affect cellular activity. So we wanted to see what was going on. So we first looked at hydrogen peroxide for a lot of reasons that we can discuss some other time, at 1 hour, which is when we saw the increase with the general ROS dye. And we looked now, instead of a general reactive oxygen species dye, we have switched to a dye that specifically labels hydrogen peroxide only. And we found that regardless of what we did, either at 200 or 500, at 1 hour, there is no change in the level of hydrogen peroxide. just in case there was a delay, because remember we said that there was an enzymatic reaction. We also looked 2 hours post-injury, thinking maybe we just didn't find it, and nope, we saw the same lack of effect from weak magnetic field exposure, though we did notice that this peak of hydrogen peroxide now appeared to be gone. So the weak magnetic fields, at least in planaria, are not affecting hydrogen peroxide levels after injury. Alternatively, we next looked at superoxide specifically with a superoxide specific dye. Again, we looked at 1 hour. And what we see is that there is superoxide at the wound site, and that 200 microtesla is able to inhibit this superoxide at the wound site, but that at 500 microtesla, there's no effect at 1 hour. But if we then look later at 2 hours, we see that there is an increase in the overall amount of, if we look in the middle where the controls are, the peak of superoxide signaling seems to occur around 2 hours after injury, which is an hour later than the hydrogen peroxide. And here now we see 200 is still able to decrease. The 200 microtesla exposure is able to decrease the amount of superoxide at the wound site. And now we see 500 microtesla able to significantly increase the amount of superoxide at the wound site. So this data suggests that weak magnetic fields are specifically affecting, excuse me, injury-induced superoxide accumulation. So if we look at the signaling pathways downstream of ROS, we see that in addition to superoxide being converted into hydrogen peroxide by that SOD enzyme that we talked about, so the superoxide upstream of hydrogen peroxide, which is not the kinetics that we are seeing. We're seeing the reverse. There's a peak of hydrogen peroxide, then a peak of superoxide. So if we look into the literature, what we see is that in addition to superoxide conversion enzymatically this way, there is also a hydrogen peroxide independent superoxide signaling through perioxynitrate, which is made when superoxide interacts with nitric oxide, or NO. And interestingly, the superoxide to perioxynitrate is much more energetically favored. It doesn't require an enzyme than the direction from superoxide to hydrogen peroxide. So we went to look at that briefly. We're still in the process of looking at this. In further support of our findings, what we find is now we're outside the realm of weak magnetic fields. We've taken them out as the weak magnetic field exposure out of variable, and we do them, quote, traditionally in an incubator, which has all different varying weak magnetic fields strengths, which is a little disturbing. But anyway, okay. What we find when we are not experimentally controlling weak magnetic fields, let's put it that way, we found that hydrogen peroxide is peaking at an hour and then reduces, and that superoxide, while present at an hour, peaks at 2 hours. And if we look at a dye that specifically labels perioxynitrate, we see the same thing that we saw in superoxide. There is some at the wound at 1 hour, but there is a peak of perioxynitrite at 2 hours. This is the quantification for it. And this suggests that hydrogen peroxide has an earlier role after injury. One of the things that we suspect is that it's not only helping with wound healing, but that maybe it's working in a sort of how to initiate a regenerative process, but that the superoxide perioxynitrite signaling has a later role in mediating the weak magnetic field effects. So later on, superoxide signaling appears to be maybe helping as a propagation signal to keep ROS signaling on during regeneration. And it's this latter part that the weak magnetic fields appear to be affecting rather than the hydrogen peroxide part. Interestingly, just quickly, we found that if we sort of genetically upregulated superoxide during injury, that we could really play around with the amount of blastema that forms. So if we knock out that SOD enzyme, the one that converts superoxide into hydrogen peroxide. If we get rid of that enzyme, this is going to result in an endogenous increase in the amount of ROS, specifically superoxide, which is sort of what we see here. We're doing RNA interference to this SOD, basically knocking it out, so it doesn't function, and then we get a big increase in ROS. And what we see is, one, if we increase superoxide levels, we are able to rescue normal regeneration when you expose to 200 microtesla. But interestingly, if we knock out this SOD, which leads to more superoxide, even in controls at 45 microtesla that normally look, control-ish because they're normal, we get a significantly larger than normal blastema, which is something that we saw with 500 microtesla. So these data seem to confirm that superoxide signaling is important for regeneration and that the weak magnetic fields are affecting stem cell activities by altering superoxide levels. Okay, so I threw a lot at you. To cap up, our data outlined an ROS signaling pathway in planaria that's required for stem cell mediated regeneration. It does it by increasing HSP70 expression, which leads to increased stem cell division and increased tissue growth. Weak magnetic fields, we found, are able to alter regeneration by manipulating ROS levels, specifically superoxide, through superoxide peroxide nitrite signaling, we think. And this occurs definitely in a field strength dependent manner, consistent with spin dynamics and the radical pair mechanism, where different field strengths can drive both gain of function and loss of function in terms of tissue growth due to either increased or decreased levels of free radicals, which is super cool because it suggests that weak magnetic fields might be a potential tool to manipulate stem cells and tissue growth. So we could use it to increase growth in regenerative medicine contexts and also maybe non-invasively decrease growth in the case of tumors. Not going to talk about it, but just as a quick teaser, I had told you that ROS signaling is also required for wound healing, even without regeneration, just at lower levels, lower threshold levels than required for regeneration, which is why our regenerative worms are still able to seal their wounds and not die. But what we found basically is that there are separate ROS signaling pathways that regulate wound healing versus regeneration, with really high levels of ROS inhibiting regeneration, wound healing, and lower levels of ROS regulating wound healing, so you can also play around, basically the point here is you can play around with ROS levels and also affect wound healing kinetics. So this could have interesting implications for like diabetics with diabetic ulcers, that sort of thing, sort of chronic wounds, which is a severe problem worldwide. Even though we haven't tested weak magnetic fields in wound closure, it suggests that weak magnetic fields, sort of their therapeutic potential, could be both broader and way more nuanced than just regulating tissue growth. So thank you for letting me talk so long. I still, even now, don't have an updated post-COVID picture of my lab. So here's what it looked like in 2019. I'd like to thank all the current students, my graduate student, Luke Kenzie, who did a lot of this work, especially the superoxide, my Beane Lab alumni, Alana Van Heusen, who's a postdoc at St. Jude's now, and an undergrad, Jacob Morton, who went through and is now a doctor of all things, who were important in driving this work, and our collaborators, sort of Frank Barnes and Christoph Simon at Calgary. So, and of course, our funding. We've been very fortunate. Thank you very much.
Thank you, Wendy, so much. And so you started with a bang our session 2, which is emerging potential evidence of quantum phenomena in biology. I just want to say that I am a fan of your data, and you have like super strong evidence that ROS is being affected by magnetic fields in a way consistent with the quantum property of electron spin. So thanks for taking like a lot of time.
Sorry.
And there are questions from the audience.
It's not working. There we are. Hello, it's Paul Davies here. I'm going to talk about these worms tomorrow, but in connection with a collaboration with Mike Levin at University, who tells an electrical story. So
yes,
it's great to hear the magnetic story. But I've just got a couple of questions. About 50 years ago, the California crazies like to say that magnets can improve wound healing. Maybe there was something in it after all. And I do remember at about the same time, people saying if you pour hydrogen peroxide onto a cut, it seals up very rapidly. Is any of that relevant to your work?
I think that's a great question. I do try to stay away from the wearing magnets aspect, just because I like to continue to get federal funding. But yeah, maybe it's not as crazy as we thought. I think that definitely has relationship to this. One of the things that this data suggests is, because it's well known that flooding wounds with hydrogen peroxide is really good, right? For making a clean wound that's going to heal, but we don't regenerate very well. So one of the things that what our data is telling us is that we need to have a better understanding of what is actually going on in terms of how we're controlling ROS, which ROS signaling it is, and when it occurs time-wise, so that if we continue using the protocols that we have, the levels at which hydrogen peroxide is used to clean wounds are prohibitive for regeneration. So if we're going to try to get rid of scarring, we're going to try to promote missing tissue, we might have to rethink, how we use the tools that we currently have, or maybe when. It's okay to use high early on within a certain window, but if we use it afterwards, it's going to interfere with regenerative. It's a little too soon for us to really sort of know definitively, right? If I had a wound, I'd probably still throw hydrogen peroxide on it. But yeah.
Hi, I have a question. Sergio Ferreira here. So very nice talk, Wendy. I was wondering, I realized that planaria are your favorite organism, but I was actually wondering if you could tell us a little bit about the effects of weak magnetic fields on mammalian cells, or either stem cells or non-stem cells. I was thinking of the fact that, for example, many physiological processes actually not only wound healing, but they actually use ROS, like fighting infection when neutrophils release ROS, neuronal communication. ROSs are essential mediators of signaling at synapses. So what is known about these effects?
What's known is not too much more than what you just said. So basically, I would say two things. If you really want to study the role of ROS in tissue, like sort of weak magnetic fields, quantum phenomenon, that kind of thing, control of ROS in systems, I would not choose planaria. Ever. I would choose a different system, but this is the one I started with, and I'm not going to change at this point in my career. But in mammalian systems specifically, host defense, sort of immunological, is probably the biggest other area that people are really interested in it. There is a large component of ROS signaling that is important during embryogenesis that could have important connotations for IVF, in vitro fertilization. So a lot of in vitro fertilization, they have very poor retention. Like, we're starting to increase the number of embryos that survive, but we don't really know why so many aren't surviving. Maybe there are differences in the magnetic fields and the equipment in the lab, who knows? I mean, I'm not saying these things are true, but they're things that we need to look at. What we do know from the literature that has been put out suggests that we really need to be investigating weak magnetic fields, because what they could potentially do is pretty mind-boggling, though I think it's going to be a while before we get there. I do know that there are some studies that have just started trying to look at altering immunological disorders, specifically with neutrophils, by exposing entire, can't remember if it was rats or mice, studies in progress. It's not published. So I'm not really going to talk about the results, but they are testing this to see if you could get it in mammalian cells. I mean, in the organism. The thing is that most of the data to date has been in cell culture, because honestly, that's tons easier to work with than an entire organism. But we are now beginning to realize that that we need to look at the entire organism. Again, I know I'm taking too much time, but just briefly, I just want to point out that like what Mike Levin sees with bioelectrical signaling, where you mess around with ion channels and you think, well, you're going to mess up everything in the body because everything in the body works on ion channels, which is true. He's able to knock out one and get very specific results. In disclosure, conflict of interest, he was my postdoctoral mentor. What it is sort of the same thing with wound healing. We can affect regeneration in planarias because the level of ROS that's required is so much higher than for wound healing. So even though ROS is affecting a lot of things in the animal, we're only ducking down levels of the thing that needs the most. So that's how we can get specific results. So it's going to take a combination of understanding different threshold levels of ROS in different tissues and situations, along with understanding what the weak magnetic fields are doing to the different ROS signaling pathways.
And I just want to make a comment here. I'm dying to make a comment. Again, just not a biologist, just a physicist. If I had to bet, I would say that a lot of those magnetic field effects should be cell type agnostic.
Mm-hmm.
I don't know how Wendy feels about that, but and also the fact that many people in the spin community actually think that some of the effects that Mike Levin might be seeing might be magnetic field mediated via spins, in a way consistent with spins as well. Just
I'm not sure I believe in complete agnosticism in terms of weak magnetic fields on cells, because there is some really cool data being done on cells communicating through radio frequencies. Oh, I'm sorry, I gotta stop. Yes.
Go on.
Okay, no, there's some really interesting data on how we get fields of tissue to react in real-time kinetics to external information. So like in an organ, in an epithelia, if you're actually going to transduce a signal, it's the same problem we have with the nervous system, right? If you're going to wait for a protein to be expressed and stuff, it's kind of slow, right? There are some really interesting data just coming out about maybe the potential of cells using radio frequencies basically to keep in touch with surrounding cells in a group dynamic. So I'm not sure I'm totally sold on agnostic to every cell, but I am totally sold on the fact that I think you can get cellular or tissue specific responses given different field strengths, different timing of applying field strengths, and how you apply them, right? Yeah.
And we have one last question.
Yeah. Okay. So thank you for the talk. It was very interesting. So my name is Marcelo. I'm a physicist from the Federal University here. And as a physicist, what really caught my attention in your talk was the fact that you have one response with a small field, then you increase the field, you have another response, and then you increase more and you have another response. Oh, that typically, from my point of view, if I had to bet, or if I had to go into this, I would say that means that it depends on the potential. It depends on the coupling between the electrons in your molecules. It depends on how they couple.
Okay.
That would be my bet. So then my question is, do you have any idea about how the quantum state is there, how the coupling, or the potential at least, how it goes?
No. I am dying to know. I've got sort of a dual personality going on, because I really think that what we are seeing is cells that are not, quote, specialized. Those stem cells are definitely specialized. So I don't know why I'm saying that. But other cells than previously recognized, able to sense quantum phenomena and make use of it, right? However, if we're going to be really honest, I love my data, I'm proud of it, but it doesn't actually, it is consistent with quantum phenomenon, but it does not actually demonstrate. We have not actually shown a change in electron state in our system for every single reason you can think of how complicated that's going to be. It's something that we really want to do, even though I think that it's worth doing, because I think it will show that we really are making use of quantum phenomenon in the cell. However, there's a side of me that is a developmental biologist that doesn't care, right? If I can figure out and I know Clarice is going, don't do this to me, right? But if I can show that weak magnetic fields are affecting ROS in a predictable fashion and are making these changes, and I understand what's going on, I can use it as a therapy. There are drugs that we use now that we're not exactly sure what the mechanism of action is, right? So therapeutically, I think this is worth going, even if I can never figure out what the electron coupling actually is or is not happening.
And can I make another comment? Sorry, Wendy.
Please, because the biologist should not be talking about physics stuff.
Okay. And again, I'm just an experimental physicist. Like, you should talk to Daniel. Daniel is the theoretical physicist here. But the fact that what Wendy sees is not monotonically increasing or decreasing with strength is actually positive. That's exactly those things. The spin dynamics of certain radicals can be modeled, and usually are relatively well understood. That one sees with magnetosensitivity as a function of magnetic field strength is not monotonic.
It is actually a really big deal. I don't know if it's a big deal on the physics side, but it is that's a really huge point from the biology side. Because honestly, when we got this data, I made the students do it over because I'm a biologist, and I think in terms of dose responses, right? Where there's all different type of growth response curves, but at some point, a little gives you a little effect and more gives you more effect, and then you either plateau or kill them or whatever. There's all different types of curves, but you never see this switching from on, off, on, off. And it was so radically odd to us that I didn't believe the data, and I made them redo it probably more than they needed to. Because I didn't, and then, I told our physics collaborator, and they're like, great. And I'm like, what are you talking about? It's not working. So I think that is large evidence that at least what's going on probably has, the quantum input. At least that's my feeling.
I would like one more time, thanks to Wendy for the amazing presentation.
Thank you very much, everyone. I appreciate being a part of this.